This conference will now be recorded.
Good evening, everyone, and welcome to today's webinar, Subcellular View of Islets to Understand Type 2 Diabetes. I am Afifa, your host for this session, and it's a pleasure to have all of you here today. This event is brought to you by Biosetup Life Sciences, a hub for scientific research and learning. We are an ISO 9001:2015 certified organization committed to supporting aspiring students in life sciences through workshops, certification courses, and career counseling. We are honored to have the founder of Biosetup Life Sciences, Mr. R. Das sir, whose vision has driven our mission to elevate students' careers. I'd also like to acknowledge our coordinator, Nabamita ma'am, for her efforts in organizing this event. Now it's my privilege to introduce our keynote speaker, Dr. Nikhil R. Gandasi. Dr. Gandasi is a renowned researcher in the field of diabetes and insulin secretion. After completing his education in biochemistry at the University of Mysore, he pursued his PhD in Sweden, where his groundbreaking work on insulin granules earned him international recognition. Currently, as an assistant professor at IISc, he is leading research to uncover how islets environments affect type 2 diabetes. His work has received numerous awards, including the Young Investigator Award and EFSD Rising Star Award. We are honored to have Dr. Gandasi with us today. Please join me in welcoming him. Over to you, sir. Thank you so much.
Okay, hopefully you can see my screen.
Yes, sir, we can see it.
Okay, perfect. Good. Now let me go to full screen view. So, okay, I actually don't know what is the background you come from. I try to go maybe overall, but then if it is too complicated, please let me know and you can interrupt me whenever you want and ask me questions. Yeah. So when you have a meal, there is an increase in blood glucose. This means that this glucose is metabolized by the pancreas, and this pancreas secretes hormones like glucagon, insulin, etc. Insulin is something that we study extensively, and that once secreted would take up help in taking up blood glucose in the peripheral tissues like fat tissue, muscle tissue, liver tissue, etc. And during diabetes, there are two problems that you generally face. One is insulin resistance, the other is islet cell dysfunction. So, insulin resistance means that the insulin is secreted, but then it cannot act. So, it keeps the blood glucose very high all through, which means that all the peripheral tissues doesn't see blood glucose. So, in compensation to this, there is an increase in islet cell activity, which means the more and more insulin is secreted, which means at some point of time these insulin secreting cells give up and they undergo apoptosis, which means there is a dysfunction of these cells. So we mostly focus on islet cell dysfunction. Most of my previous work has been focused on that. So I've been speaking about islet. What is an islet? So pancreas is the size of a small fist, and this has a large part of exocrine tissue, which is in yellow that you see here, and some parts that you see in green, which are islets of Langerhans, which you see here. These islets of Langerhans as well have three different type of cells: insulin secreting beta cells, glucagon secreting alpha cells, and somatostatin secreting delta cells. So we mostly work on these islets to understand type 2 diabetes and type 1 diabetes at IIC. So we study underlying mechanisms of how insulin is secreted, what are the driving forces of it, what are the other complications, and can we have mitigation strategies, in the sense treatment strategies to treat diabetes. So that is the reason I have chosen the topic as subcellular view, trying to study these individual cells to understand diabetes. So with this context, I'll give you a little bit more insight into single beta cells. So these single beta cells, once the blood glucose increases, there is a GLUT transporter on the membrane of these cells, and they take up glucose. When they take up glucose, there is glycolysis and Krebs cycle and things like that, which you might have heard of in the previous classes. So those generate ATP, and these ATP closes the KATP channels, and there's always a charge ratio that's maintained between the positive and negative charges, and this charges are lost, which means that the calcium channels open in so that the calcium can come in. So this calcium coming in means that these large dense core vesicles, sorry for that, large dense core vesicles that you see here in gray, those are filled with insulin granules, and they fuse to the plasma membrane to release the content. So it's a calcium sensitive process. It's called regulated exocytosis. So we study this using various microscopy techniques like confocal microscope, which you might know of, where the optical sections of a cell are taken, or turf microscope, where you can visualize 100 nanometer below the plasma membrane.
Excuse me, sir.
Yes.
Sir, the slide is stucking, sir, not move.
Okay. So which slide is it stuck in?
Sir,
It
from
says
first slide.
subcellular organization.
Okay, that's weird. So do you see my screen now?
Yes, sir, we can see it, but it was stuck on the introduction slide.
It is still stuck there, is it?
Yes, sir.
Okay. Let me try to.
And now it is moving. Now it is moving.
Now is it moving? Okay. Let me just see. So do you see something moving or nothing?
I think this is second slide. Diabetes and islets hormones.
Correct. And then do you see pancreas here?
Yes, sir. I can see the whole slide.
So do you see something now? Something in green at the bottom?
It is still on the second slide only.
Okay. I'll try to do this in a little
Yeah,
bit of.
now it is the third. Now it is on next slide.
I think there's
It
a
is
delay
moving
there.
one by one. I think the
I'll
slide
just
is
stop
moving one by
sharing and I'll try to share the screen as a whole window.
Yeah, yeah.
Let's see,
Okay.
maybe it works better. Do you see my screen now?
one. Yes, sir.
Yes, sir.
Okay. And hopefully you see it now.
Yes, sir.
Okay, perfect. So speaking about pancreas, and in the pancreas, there are small little islands of cells which are called islets of Langerhans. And these islets of Langerhans, they are made up of insulin secreting beta cells, glucagon secreting alpha cells, and somatostatin secreting delta cells. Please let me know if you have questions in between. Yeah. So we try to work on these islets to find out what happens in type 1 and type 2 diabetes, with an idea to see what is the underlying mechanisms which is taking care of insulin and glucagon secretion, etc., and what are the treatment strategies that can be thought of when we say about diabetes. So I was explaining you about glucose transport, and an increase in glucose that leads to increase in ATP because of glycolysis and Krebs cycle and things like that. And there is KATP channels that close. So there is a charge ratio that's maintained between positive and negative charges. And this, when it is not in equilibrium, that is when depolarization occurs. And then there's calcium that comes in. This calcium means that these large core vesicles that is present in pink there, those fuse to the plasma membrane to release insulin. So this is a regulated exocytosis because it's driven by calcium. Each one of these vesicles, they fuse to the membrane to release insulin through exocytosis. So I was telling you about different strategies that we use to study these islets and also single cells. One of them is confocal microscopy, where you can take optical sections of a cell to see where these individual granules that are there in black, as you see here, are present. So the other is something called turf microscopy. In turf microscopy, you only see vesicles which are just below the plasma membrane of the cell. It covers like 100 nanometer below the plasma membrane of the cell. So these are very specific because it gives a very good signal to noise. And the other is very generally used fluorescence microscopy, which is epifluorescence, where you see the entire cell. I'll show you images from confocal and turf microscopy. What you could probably appreciate is that these individual granules, which are fluorescently tagged, you see them as dots, and you only see them at the bottom of the cell. You don't see them from the upper parts. You don't see them from here or here. You just see them from here, which is a huge advantage because our process occurs at the plasma membrane and we can visualize what is happening at the plasma membrane of the cell. Whereas, let's say in confocal microscopy, you would see individual vesicles which are there in the periphery of the cell, and then you would probably see something in the middle here. It gives a lot of noise in this case, which is not really beneficial because you can't really track single vesicles that you see here, whereas you can do that very well in this case here. So this is again a turf microscopy image, and you can see individual granules which is like 100 nanometer below the plasma membrane of the cell, visualized using turf microscopy. And as it happens in the body, we stimulate them with glucose. We take it out, we put it on the cover slip, and we stimulate them with glucose, as you can see here in the cartoon. So when you do that, and this place. Okay, I'm sorry. Somehow the movie is not playing. As you can see here, there are vesicles which are moving around, and when these vesicles move, after a while, you start seeing them brighten up and then disappear. You can see a lot of them right now. So this is in fact what is exocytosis. They're coming to the membrane, they're in the turf field, they're increasing in brightness, which is to say that we're very close to the membrane. There's a change in pH and they disappear. So you see these individual events like this. This is an image strip which shows individual exocytotic events. So the opposite of this is docking. When these vesicles come and fuse here, this is called docking. And then when it releases the content, it's called exocytosis. Just to show you a difference, this is exocytosis, and this is from the vesicles coming to the membrane, which is called docking. So these were all exocytotic events that I showed. The opposite of this is docking, where nothing is visualized before, and then they come to the membrane, and then they stick there, right? With this sort of introduction, I'll go into the data. We had access to human beta cells, and we tried to have fluorescently tagged insulin granules. And here are the insulin granules that you see there. And you see a large density of dot granules. So these are the individual dots, are the dot granules that you see there. And we stimulate them with glucose, like I showed in previous two slides. And when you see that, there's a large number of exocytotic events that you see. And that means that with every donor that we have, every patient that we have, we can try to visualize it by saying how many dots are here and how many of them underwent exocytosis, which means it gives a very nice correlation between the density of dot granules, what's the total number here, versus how many of them underwent exocytosis. What you could probably appreciate is that there is a small fraction of granules below here which comes from type 2 diabetic individuals, which means there's low density of dot granules. We tried to take advantage of this and try to see what happens in case of type 2 diabetic individuals, and what we see is lower density of dot granules. So each dot here is a donor, or if you want, you can call it a patient. So these donors, all of them, they have less number of dot granules like you see here, whereas the non-diabetic individuals, they have more dot granules, like you can also see it in the image there. So what does this mean? So normally, if you see the peripheral blood, let's say I take blood from any one of you, and if you're normal, you get a biphasic response immediately after you had a meal, which means that for 2 hours, there will be an increase in insulin secretion, and there's a slow increase, which means that all the glucose that is in the blood from one meal to the other is completely metabolized during this time in a normal individual. Whereas in a type 2 diabetic individual, there is no biphasic response. There's no first peak here or the second peak, which is how this looks like in the peripheral blood. So what does this mean to the individual islet that I'm speaking about? What does this mean to single cells of the islet, the green cells of the islet that you see there? So these green cells also show a very nice biphasic response, like you can see a peak there and then a short sort of small peaks there until the glucose is metabolized. In a type 2 diabetic individual, what you see is that there's no peaks. You see very short peaks there, which means that the density of dock granules is fewer, like I showed you 2 slides ago. And that means that the density has to deplete, and that depletion is not happening, which is what is the problem in case of type 2 diabetes. So I was trying to explain you regarding the phasic response, like the first phase and the second phase and things like that. I also told you about the GLUT transporters and how glucose is taken up. We started seeing if insulin secretion is decreased in type 2 diabetes, what happens? Does it take up glucose? Does it not take up glucose? Was the question that we were trying to address. During this time, we had access again to human islets. One of the students, PhD students in the lab, Pallavi, she tried to image these type 2 diabetic beta cells, and she tried to compare it with non-diabetic individuals, where she could see that the density of GLUT transporters, so this is the GLUT transporters that you see there, and the density of this, they should increase. When there's an increase in glucose, they should increase in density at the plasma membrane, which means that they can take up glucose. This is completely gone in case of a type 2 diabetic individual. You see very less number of GLUT transporters that is present in the turf field there, which also goes very well with the expression of GLUT transporters, which is also very less in type 2 diabetic individuals. So just to explain that, you see the GLUT transporters present at the membrane there. In the type 2 diabetic individuals, they can take up glucose. These hexagonal structures that you see are glucose being taken up, which means that they can drive so that the insulin granules can fuse to the plasma membrane to release insulin in the form of exocytosis. In case of type 2 diabetic individuals, there are fewer transporters that are present in the plasma membrane, which means that less of glucose can be taken up in the first place. And secondarily, there is also fewer number of granules that are present at the plasma membrane, so that also means there is reduced in insulin exocytosis. So we try to treat, we wanted to see if we can treat this condition. So what we did was we tried to develop a compound which is called fiopharbide A. So this is one of the postdocs in the lab, Dr. Saptadeepa. So she tried to do by having a compound with fiopharbide A. There she could see that we could have more GLUT transporters at the membrane. So you can see that there. And these GLUT transporters metabolize glucose much faster than very well-known drugs such as metformin, which is used to treat diabetes. So we wanted to see what happens. Why does it drive GLUT transporters? So we saw that the transporters that exist, they keep them arrested at the membrane. So which means that they're always there at the membrane. Normally, when they're there at the membrane, that once it takes glucose in, they again endocytose. They come back to the cytosol. In this case, with this drug, it always keeps at the membrane, which means that more and more glucose can be taken up, which was a very good hit for us, which means that with just targeting the GLUT transporters, we had an answer to have more and more glucose being taken up. But that doesn't mean anything for the insulin secretion, because that, I said, is driven by duct granules, and there's a decrease as well. So we want to see what happens to these duct granules and how do that gets driven. So then we started seeing the expression of various proteins, and some of the proteins that are present in docking, they're reduced in case of type 2 diabetes, like rim2a, rab3, rim1a, etc., which means that there is less secretion is what we realized. So we started seeing what makes docking so special then. So one way we can try to increase glucose transport and then still drive one part of the cell, but it doesn't mean anything for the docking. So we started to understand the mechanism of docking. So we started to go more into details of what are the different proteins involved in docking. One such protein is syntaxin. So syntaxin is a snare protein that exists at the plasma membrane. So it always has a transmembrane domain, which means that it is always present at the plasma membrane of the cell. So these are very important because whenever there is a granule coming from the cytosol to the plasma membrane, the single molecules of syntaxin, they gather together to form a cluster where a stable docking can happen. So if you see this movie here, what you can see is that there are green syntaxin molecules that you see. They come together, and then the red is the granule that you see there, and they try to attach to the red granule so that it can stabilize a docking complex. So this stabilization means that there is granule coming to the plasma membrane, and then there's syntaxin clustering that happens in parallel, which means that there is a stable docking that takes place all the time. And this stable docking is very important for the exocytosis to take place. So once this docking happens, next you see other proteins like MUNC18, MUNC13, calcium channels, all of these being recruited. So you can see the granule coming to the field here, and then you see recruitment of other proteins. After syntaxin, MUNC18, there's MUNC13, calcium channels, etc. These are very important because this means that there is exocytosis that can take place. So all this form a complex here, which means that the insulin which is present in the vesicle can do the release here. So this means that these clusters of syntaxin disappear immediately after its job is done. You can see here that there is the signal here, and it just completely goes down. And in case of non-excitotic events, it doesn't go down. It always stays the same. And weirdly enough, we also started seeing that there are lipids which are part of this, one of them being phosphatidyl serine, which also is important, which forms a stable complex here, which means the exocytosis take place. We started looking at other lipids, which are like PIP2 and things like that. We have had some hits recently. So PIP2 is one of the abundant lipids in the plasma membrane. So we were able to develop a probe for PIP2. So this probe meant that we could visualize PIP2, and whenever we see exocytotic events, you see these dots coming up here. These dots, all of this is PIP2 driven. So you see an increase in PIP2, especially at the places where there is exocytosis. So wherever there is no exocytosis, there is no PIP2. But wherever there is exocytosis, there is very high increase in PIP2 that you see, which is very nice, because which means that we can follow this and see what happens to the transporters and things like that. So until now, what I've been telling you is regarding insulin excitosis, how the granules come to the membrane and they undergo a process called docking, and how other proteins are recruited, and how that enables so that these vesicles can fuse to the plasma membrane to release insulin. So syntaxin forms the initial cluster, and then MUNC13 calcium channels are recruited much later in a process called priming, and these primed granules undergo excitosis. So there are many proteins which are part of this nanodomain. So some of these proteins are depicted here. I tried to explain more molecular mechanisms regarding syntaxin. So all of these proteins have their own molecular mechanisms, which in the interest of time, I'll not go through, but I'm just trying to get you interested to think what each one of these proteins might be doing and why it is important for type 2 diabetes at all. So to understand this better, what we tried to do is we tried to take beta cells from type 2 diabetic individuals. We saw that some of these proteins are very less, like I showed you 2 slides ago. Sorry for that. I'm going too back, I think. Yeah, yeah, there you go. So you see the decrease there. So we tried to see how we can compensate for this decrease and how that can be done. And this compensation we tried to do with by overexpression of these proteins, one of them being syntaxin. You can see that whenever we increase the expression of these proteins, there is an increase in the density of dot granules. The total number of dot granules increases, which is very nice because it has a translational potential is what this means. So also, let's say for RAB3 we saw a same effect, with RIM1 we saw a same effect, RIM2 we saw the same effect. So which is all good, which all means that overexpression of these proteins would help in reversing diabetes in the beta cells, which doesn't mean anything because it's an experimental test. Because if you try to see like 200 donors, in all of them, if you try to measure the insulin secretion and try to correlate this with some of these proteins that I mentioned, there is an inverse correlation, which means that there is less expression of these proteins, which means there is less of insulin secretion that's taking place. So one of the factors to measure how diabetic a person is something called HbA1c. HbA1c measures how glycated our RBCs are. If the RBCs have very high glycation, then the HbA1c would be very high. So HbA1c which is less than 5.6, 5.7 is definitely the person is not diabetic. Anything over 6 means that the person is heavily diabetic, so they need to be treated. So you see here that with if a person is heavily diabetic, they have less expression of many of these proteins, which all goes to show that overexpression of these proteins would be a good treatment strategy. But you can't do this in an individual because it's a very artificial setting. So now we are trying to see with some of these compounds that we have discovered, can we try to drive it to increase the expression of these proteins? And this increase, what does it do for the exocytosis to take place? So one such protein is called EPAC2. So that is already being used as a treatment strategy. So you might have heard of GLP-1 analogs. So whenever you go to a clinician, a doctor, and you're diabetic, normally they prescribe two things. One is metformin, and the other is GLP-1 analogs like Exendin 1, 9, and things like that. So these GLP-1 analogs, what do? Say they normally increase the insulin secretion. We want to see molecularly what do they do. So one such protein which is involved is called EPAC2. So this EPAC2, when it's given to a non-diabetic individual, you see EPAC here. This is how it looks like. Whenever you treat the single cells with GLP-1, there is an EPAC2 clustering that you see at the single cell level. These clusters localize very well with insulin granules, and they help in release of insulin. There's more insulin excitosis that take place. So in type 2 diabetic individuals as well, when we treat them with GLP-1, there is a clustering that you see there. But interestingly enough, there is HbA1c, which I said how diabetic a person, an individual is. So at the scale of this, you see at 5.6, a person is definitely not diabetic, whereas at a scale of 5.8 or so, the person is pre-diabetic. At a scale of 6, the person is diabetic, and as you go on, the person is more and more and more diabetic. So what we try to do is we try to treat these cells from these donors with EPAC2, in the sense we try to increase GLP-1 through analogs or GLP-1 itself. We could see that there is an increase in insulin exocytosis that we see when the people are pre-diabetic or a little bit diabetic. If they're severely diabetic, let's say more than 6.6 and so on, this drug doesn't do anything, which was a surprise because people who are severely diabetic also are given these kind of drugs today. So we need to think about what kind of strategies that we do. So our studying the mechanism of single cells is giving a very good idea of what is going on in the single cells and what drugs we can use and what drugs maybe we can't use, is what I think. So to summarize this part, there are several defects that you see at type 2 diabetes. One is with GLUT transporters, whereas very less GLUT is recruited, where we have, I think, found solutions and we know what to do and how more and more GLUT transporters can be recruited to the membrane. And then there's a process called docking, which I mentioned about. And there as well, there's a decrease in total number of dock granules that you see there, and that also means that there's less priming that takes place, whereas these proteins like EPAC2 are involved, which ultimately means that there is less exocytosis in case of type 2 diabetes. So our treatment strategies are targeting this particular step called priming, which is what I mentioned about EPAC2 and the GLUT transporter that I mentioned previously. So, but there are other cell types as well. So the other cell types in an islet include, apart from beta cells, there are alpha cells, there are delta cells. So the alpha cells that you see there are what do you call this? Purple in color, and the delta cells, which are pink in color. And these cells are also important. So we want to study these cells because I spoke a lot about insulin secretion until now. Insulin is secreted when you're in a very well fed state, but glucagon is secreted when you're in a fasting state and when you're hypoglycemic, which means that where the blood glucose is very low. And most people, in fact, die out of hypoglycemia, not hyperglycemia. So we wanted to see how we can target these purple cells or glucagon secretion. So one of the biggest challenges when we stepped into this was that we couldn't label these cells. These cells in an islet was very hard to label. So we had to find probes to label these cells. So we developed a probe which is called PPGEGFP. So we can visualize these individual alpha cells was the idea. So when we did that, we found that we could label them, and also once we labeled them, we were able to see the single cells. You can see alpha cells here, and we could also see what are the different effects in these alpha cells. What does this, because the islet, as I can see here, is an organoid which is consisting of beta cells, alpha cells, delta cells, etc., which means that they all have to be in an environment. They all have to be in a milieu to conserve these cells in a context. So in this context, what happens is when we study the alpha cells, we see that the beta or delta cells, they drive the alpha cells to a large extent. When you're in a fed state, a signal is sent from the beta cells saying that you don't have to secrete glucagon because we are not hyperglycemic, or a signal is sent when we are very low on glucose, say by delta cells saying you should secrete glucagon. All this is very important to maintain the blood glucose level. And this maintenance of blood glucose as well is not the same in case of type 2 diabetes, because the receptors that are present for signaling these, they're absent in an alpha cell in case of a type 2 diabetic individual. So we also see, therefore, that there is a decrease in glucagon secretion, which is what is depicted here, the decrease in glucagon secretion in the type 2 diabetic individual, which also means that when you try to see at these individuals, you see HbA1c, like I explained before, and this HbA1c, the red ones are diabetic here, the black ones are non-diabetic individuals. You see that the glucagon exocytosis reduces in the diabetic individuals, and this goes very well again with the density of glucagon granules present there. And as you can see, the inverse correlation between HbA1c and density of dock granules that you see there also in alpha cells. So this explains one more thing, why people do die out of hypoglycemia, because when they are not able, when they are in a state where there is not much blood glucose in type 2 diabetic individuals, there's no treatment strategies that in fact trigger glucagon secretion. Most of the triggers are in case of beta cell insulin secretion, which is not sufficient. You need to also target glucagon secretion so that people can manage their hypoglycemic state. So you might have heard of people with diabetic people, they carry chocolates with them when they feel really low, and when they are about to go to coma, they just bite a chocolate in and they're fine. Why do they do that? So that they don't enter the hypoglycemic state. That's the idea of this. So that is very critical, so that the glucagon is secreted. And this change in glucagon secretion in type 2 diabetic individual means that they need to have a higher response, which is not really the case in them, which needs to be managed as well. Then we started looking at the third cell type, which is called delta cell. You see these pink cells which are present. So these are the delta cells. They're very few in number. If you consider an islet, the total number of delta cells is less than 10% in an islet. But then these also matter. So people were thinking, why these cells matter? So we showed the signaling between alpha cells and delta cells, which means that in a hypoglycemic state, glucagon is very important, and that glucagon secretion is maintained by delta cells. And these delta cells, when we look at the number of delta cells in a type 2 diabetic individual, they are decreased, which was a very recent finding. In last year we found this, which means that the controller of alpha cells which secrete glucagon, they are also less in number, which means that the control is less of glucagon secretion, which partially explains, sorry for that, which partially explains why you see a decrease in glucagon secretion that we see in case of type 2 diabetic individuals, we believe. So this all means that these delta cells which are in green here, they control beta cells and they also control alpha cells. And there is a decrease in mass of the islet cells, and this decrease means that the glucagon goes through a counter-regulatory action, and that becomes too much in case of type 2 diabetic individuals, and that can also lead to problems both in case of type 1 and in case of type 2 diabetes. This is one of the recent findings that we published this year. So, to summarize this part, it's very important that insulin is secreted, and this insulin secretion is maintained at several leads, which is through beta cells, which is through GLUT transporters, which is through docked granules, which is through primed granules, and also at the level of exocytosis. And this means that if you treat them, which means that the insulin secretion is maintained, and if there is decrease, there's also decrease in glucagon secretion that we see in case of type 2 diabetes. So this is about insulin secretion. This is treatable is what we have found. With respect to glucagon secretion, we have found that there are problems here, but then there is no way to how to treat them, I think. So we need to work more and more towards it, and there's not much known at all in case of delta cells and somatostatin secretion. There a lot of work is required to understand them and also to find treatment strategies, which is what our lab is doing, trying to understand the individual cell types through imaging and trying to study the mechanisms, and with these mechanisms to study what are the treatment strategies that we can follow, and we can try to treat diabetes. So with this, I would like to acknowledge all the funding agencies and the research group which is involved, and ISC for this work. Thank you very much. I would be happy to take questions. Hello, am I audible?
Yes, sir. Yes, sir. You are audible.
Sure. I see there are things in the chat box. So it's mostly regarding my
Yes.
audibility, I see.
No,
So
sir.
if there
You
are
are.
specific...
It was audible. I
Okay,
think they
okay,
must be
great.
having some network issues.
Sure, sure, sure. Looking forward to questions.
Okay. Thank you so much, sir, for that insightful presentation.
Thank you.
We now have time for a few questions from our audience. If anyone have any question, please you can type them into the chat box or you can unmute your mic.
So I see one
I
question
think
in the chat box,
I
which
have one
says,
question.
is syntaxin made up of? So syntaxin is a protein which is present in many cell types, mostly secretory cell types. And the syntaxin is made up of snare domain, which keeps them, which is also a transmembrane domain, which means that they're always kept at the plasma membrane of the cell, which is like the plasma membrane is here, and always there is syntaxin that is present at the plasma membrane, which means that the vesicles can come in and stick to the plasma membrane. So these syntaxins, they form a cluster, and this cluster means that there is a stable docking process that can happen. And if this doesn't happen, there is fewer number of dock granules. You see very few granules at the plasma membrane of a cell, if that answers your question.
Yes, if anyone else have any question, please
So I see
you
questions
can
regarding
unmute your
ISC.
mic.
So with respect to dissertation projects and things like that at ISC, so it's mostly up to the individual, in the sense the PI. So we usually hire like 4 to 5 people every time whenever during dissertation. We have numerous applications. We end up, let's say, from 50 to 5. So it depends. We do extensive interview process once the people come in. Every lab has a different approach. So we are a total of 75 labs that exist. My lab is only one of them, but there are many other labs. So you have to approach the PI, and then you have to go through the process, and then you can get into dissertation, I think, if that answers your question. So there's one more question which says, is there any role in maintaining the charge of syntaxin? So yeah, syntaxin is maintained in a particular way. So it has positive charges, which helps it in a Brownian moment that takes place. So this Brownian moment is seen in individual molecules of syntaxin. When there's a cluster, this charge is still maintained as such, and this charge is completely lost when the cluster breaks down again, if that answers your question. Any more questions?
Yes, sir, we have one more question.
Sure.
Shweta Paul is asking, is there anyhow cholesterol concentration modulating the docking process?
So the cholesterol is extensively present at the plasma membrane of the cell, and this maintains the plasma membrane integrity. So we have done experiments on depletion of cholesterol. Depletion of cholesterol also means that there is less of membrane content and there is less of insulin excitosis. There is no impact on docking for this, but there's definitely impact on insulin secretion. There is less of insulin secretion that happens in this case, if that answers your question.
Yes, there is one more question.
Sure.
Neha is asking if we wish to target to exocytosis promoting proteins, is there possibility by upregulating one we downregulate others? So are those proteins a potential target for drug discovery?
Yes, yes. So that's the idea. So the drug discovery process is right now completely dependent on KDP channels, where sulfonylureas is something you might have heard. Many people might be given sulfonylureas as well if a person is diabetic, maybe at your home or something like that. And the other is the one that I was speaking about, GLP-1 analogs. These are the very extensively used treatment strategies. So these proteins and new targets that we are trying to identify means that we can try to find a compound and we can try to see docking itself can be a process, and insulin granule docking means that there can be more excitosis, which is one of the approaches that can be taken to increase insulin secretion. That's the whole idea of it, but we have found a pathway for it, but we still don't know how we can increase it with activator or something like that we don't know yet. We are testing several of them, but we still don't know where it can fall, I think.
Okay, sir. So, any more questions, dear participants? Please, if anyone have any question, they can type them now. Okay. Ahmed is asking, how does change in mass of cells indicate the presence of counter-regulation mechanism for glucagon?
So, I think in the first few slides, I tried to explain regarding how there is overcompensation that happens. When there is a decrease in insulin secretion, there's always your cells, it tries to say that, well, I can't have insulin. Let's say your muscle cells or so says, I'm not seeing any glucose, which means that these cells try to overproduce the same number of cells. They try to produce more and more and more insulin, which means that at some point of time, they are completely stressed. The endoplasmic reticulum stress kicks in, which means that the cells start dying. So that is where the total density of cells decreases. So what happens is the beta cells are decreasing, but the alpha cells remain same, which means that the counter regulation between beta cells and alpha cells is not maintained at all. So there is no ratio. So the amount of insulin producing cells, amount of glucagon producing cells, they're not maintained in a ratio, which means that the counter regulation is completely lost, which is one of the factors in case of type 2 diabetes, if that answers your question.
Okay, sir. Rohit Kumar is asking, how can we get the record? Okay, okay, this is for me. Sorry, this is for me. So, how, how can this subcellular view help in discovery of long-term regulation and treatment of type 2 diabetes?
um, So I think there are many different treatment strategies that we have found out for diabetes. One is to manage glucose. That is the easiest usually. So that's why people exercise to manage glucose and things like that. And then there are targets such as metformin, which also can help in glucose transport in other peripheral cell types and things like that. And there are like other ones which I told about sulfonylureas and things like that. These are not really solving the problem, right? So we are just treating the symptom. We're just treating less secretion of insulin, that's it, right? So we are not really we are treating a symptom, but we are not treating the disorder itself. So the idea is trying to understand what are the changes that happens in the disorder, then to see if we can target and change things in the disorder itself. So that's the overall sort of overarching thought there. But we need a lot more work to reach there, I think. Right now, all the treatment strategies are mostly managing the symptoms, nothing regarding reversal.
Okay, sir. So Iram Khan is saying, in your research, what molecular determinants have been most significant in disrupting the insulin secretory process in beta cell of type 2 diabetes patients, and how do these determinants interact with each other to affect beta cell function?
So we have a transcription factor called TCF7L2, which has some mutations in some people. So these mutations mean that there is less expression of syntaxin in the first place, which is seen in case of many of the type 2 diabetic individuals. So that's why the artificial process where we did of increase in expression of syntaxin. So there are many other proteins like this, like RIM1, RIM2, calcium channels, EPAC2, and things like that. So all these expressions can be increased. But one of the things to manage it is many of our people, you don't know what are the different variants that you have. You don't know if you have any mutations or SNPs and things like that. Maybe you might have heard of, I didn't speak about this at all, the genetics part of diabetes, where the person father has diabetes and the daughter or the son also has diabetes. So this family thing mainly occurs because you have the risk genes. TCF7L2 is one of them. And these risk genes, if you find it out, I mean, if you get yourself sequenced, if you have heard of 24andMe, that's one of the companies. I'm not marketing anything, but just telling you. So you can get yourself sequenced. You know that these are the risk factors you have in yourself, which means that you will definitely get diabetes, which means it's easy to maintain early. Or people seek genetic counseling, which is one of the approaches. But this is the only way we know of. Can we treat this? Is what we are looking at, I think.
Okay, sir. I think we have covered almost all the questions. So,
Okay.
as we conclude today's session, I want to extend a heartfelt thank you to Dr. Gandasi for sharing his expertise and to all of you for your active participation. Thank you, everyone. We appreciate you being here. And I am sharing a feedback form link in the chat box. Please make sure to fill it. Uh, Please make sure to fill it. So, upon submitting the form, you will receive a certificate of participation. So please make sure to submit the form before you leave the meeting. We hope this webinar has provided you with valuable insights to guide your career journey. Please stay connected with Biosetup Life Sciences for more such enriching opportunities. Have a great day ahead. Thank you so much.
Just a request. Can you share the feedback form with me as well, please?
Yes, sir, I will. I will share it.
Sure.
I
Sure.
will share
Thank you.
the response with you.
Yeah,
Okay,
and
sir.
if any of the students want to approach and want any guidance in terms of career and things like that, happy to give that. All the very
Okay, sir.
best. Thank you. Bye bye.
Thank you so much.
Thank you so much, sir.